In Unit 4, we'll cover Chapter 5, the working cell. In this chapter, we'll discuss enzymes, membranes, and the transporting of materials in and out of cells across those membranes. We will use the nervous system cell known as the neuron as a good example of different You should remember from last week that the plasma membrane, or cell membrane, is made of phospholipid bilayers. The phosphate heads have polarity, which gives them their hydrophilic sides. The fatty acid tails face inside the inner membranes, and that gives the hydrophobic region. Together, along with the proteins and other substances attached in and on the membrane, our membranes become selectively permeable, meaning they can be choosy about what gets through or not. Now, the membrane itself has pores between those phospholipids, and many small things are able to sneak through. But most things, we have a very finite sort of choosiness about what can get through. Recall that that is called selective permeability. We also have recognition and receptors. Both are important in telling or signaling the cell to do something, like let a substance in or letting a substance out. The transporting of molecules across or through a membrane can be done through either passive or active processes. Passive implies no energy is being expended. Therefore, it's relying upon the random motion of molecules. In active, energy is actually expended to move these molecules, typically across a membrane and often what we call against a gradient. Now, let's start with passive. One type of passive is known as diffusion. Diffusion is a random process, does not require energy, and comes in two types. It could be simple, which means there's no proteins or carriers involved, or facilitated, in which we often have proteins or carriers that help the molecules through a membrane. Remember, both of these are passive, so no energy is expended. Let's look at an example of simple diffusion. In the top series of diagram, starting on the left-hand side, you'll see that the molecules of dye are in high concentration on one side and low or no concentration on the other. The side that is high in concentration in these dye molecules is said to be hypertonic, and the side without the dye molecules is hypotonic. Due to molecular motion, the molecules will passively move through the membrane from hypertonic to hypotonic. This will occur, as you see in the series across the top row, until an equilibrium is reached. An equilibrium is known as isotonic. Now in the series on the bottom, you see two different molecules. Each has its own gradient or concentration difference. We say in diffusion that the molecules are moving down their gradient. So you need to count where the molecules are highest and compare that to its lowest to know which direction each will be moving. In facilitated diffusion, recall that this is passive, but it's one in which we need a doorway. These doorways are proteins embedded in the membranes, and these doors are passive in that they open and close as long as there's some general recognition that entrance is needed. In the example, you see glucose sitting outside of a cell, and a cell requiring or needing that energy to be brought in. Specific carrier proteins, also called channel proteins, will open and close, allowing the glucose to pass through the membrane. Now, another type of passive movement is that of water, and that is known as osmosis. Osmosis is moving down its gradient, just like those molecules were in diffusion, but the gradient might not always be exactly the same. If you think about that, a solution that has more solutes and less water, solvent, would be hypertonic. A solution that has very low solutes and lots of water or solvent would be hypotonic. And of course, isotonic is when it's equal. In the illustration on the bottom, you see what happens when a cell or some structure with only a thin membrane, meaning no cell wall, is placed in these different solutions. If a cell is put in a hypotonic solution, the water is in higher concentration outside and therefore moves into that cell. If that cell has no ability to regulate the water fast enough, it'll explode. That's called lysed. In an isotonic solution, well, the net exchange is equal, so the cell will be normal. But in a hypertonic solution, there's less water outside the cell, so water is drawn out and a pickling kind of event takes place. The cell will look shriveled. This is called crenate. Here's an example where we'll couple diffusion and osmosis. So this diagram is something similar you're going to be doing in lab. We have a simulated cell made out of dialysis tubing. Why is that important? It's made of a semi-permeable membrane, so it will allow some things to move in. Water and salt are going to be the two things that are in our example. In the lower right-hand corner, I've given you the setup of this potential experiment. In the outside solution, in the beaker, is a 10% salt solution. So remember, that is 10% salt and 90% water. Inside the bag is a 2% salt solution, so 92% salt and 98% water. Which way will diffusion take place and which way will osmosis take place? Both salt and water will be moving down their gradients, meaning from high to low. So the salt is higher outside in the beaker at 10% and only 2% inside the cell. That means salt will want to move into the bag. Water, on the other hand, is only 90% inside the bag and 98% outside. So the net movement of water will be into the bag. Let's change up that experiment slightly and put a hypotonic solution in the beaker outside of the cell. In this case, we're going to have the beaker water being 100% pure water, meaning zero salt. Therefore, inside the bag, at its 2% salt solution, has a higher solute concentration. That means the salt inside will want to move out through diffusion. The water, on the other hand, is the opposite. We have 100% water on the outside and only 98% inside the bag. Therefore, water will want to In active transport, we have to expend energy in the form of ATP. Think of a doorway in which you must use energy to open and close it. Many of these proteins embedded in membranes have a high degree of what we call specificity, meaning they're choosy. You might have to have the exact right shape as well as energy before you'll be allowed to enter or exit this cell. Active transport proteins allow our cells to have the greatest level of semi-permeable choosiness. Imagine needing to get something in bulk, like large quantities in very quickly, or perhaps a very large packet. This is called endocytosis, and things can be brought in through the membrane by physically wrapping part of the membrane around the substances being brought in. You'll notice in the diagram it's showing you phago and pinocytosis, meaning eating or drinking. So these could be solid particles or more of a liquid variety. I often sort of joke that this is how you get in if you're too big to get through the doorway. Well, these are really important in our white blood cells, where they phagocytize or engulf particles such as bacteria and other foreign invaders in an attempt to neutralize them. Imagine the opposite. You've built some large molecules and you need to export them or get them out of the cell. You do kind of the reverse of endocytosis. You have to have it wrapped in a membrane, and that would be done at the Golgi. Those vesicles go to the outer plasma membrane, fuse, and then spill out their contents. This is a really important way to move things like hormones and digestive enzymes and even neurotransmitters that are inside of our cells exported out to the surrounding fluids. We'd like to showcase the neuron, the functional cell of the nervous system, to show you some of these basic principles of transporting in and out of these cells. Let's start by looking at the neuron structure. All messages are meant to be signaled down and through these neurons. Therefore, their structure is really about sending information. All information is picked up through dendrites. These are small projections from the main body that receive information and conduct it to the cell body, or the main part of the cell. In this diagram, you can see the cell body has the nucleus, and this is where most all of the organelles are kept. Information flows from the dendrites to the cell body and then out the axon, a single thick projection. At the end of the axons, it branches yet again to further communicate into other structures. At the end of the axons are small axon endings in bulbous structures that we refer to as boutons. The conducting of messages through our neurons really is an electrical impulse, at least at first, as the message moves from the dendrites to the cell body and down the axons. That is a guiding principle of passive and active transport. We'll look at that here in a sec. But at the very end of that neuron, remember those ends boutons, also known as synaptic knobs, we have a type of exocytosis taking place in which neurotransmitters are released from that knob or bouton into a gap or space between one cell and the next cell. Those neurotransmitters move across that gap and stimulate the next cell or next structure in line. This is how our cells communicate through the nervous system. The function of the neuron is really to send a message, but in doing so, it has to do some diffusion. Specifically, we'll be looking at sodium and potassium and how they move in and out of the neuron to create that electrical impulse or that message that moves down both the axon and the dendrite. Let's zoom in and look at the actual phospholipid bilayer of, well, in this example, an axon. But this membrane would look the same if we were looking at it up there in the dendrites too. There are lots and lots of channel proteins embedded in that phospholipid bilayer, some that are open all the time, but the ones we want to be most focused on are the gated channels. These are closed and only open when told or stimulated to do so. Imagine if you had a gated channel protein for sodium. Sodium would only be allowed in if the gated ion channel was open. And the same for potassium. But remember diffusion. Everything moves down a gradient, or areas of high concentration to low. So in this example, if the gated channels opened, and if there were specific channels for sodium and specific channels for potassium, each will move down its own gradient once the gate is open. If we count up the number of molecules, the passive diffusion or facilitated diffusion taking place would be sodium moving into the cell and potassium leaving the So when a neuron is at rest, meaning it's not doing any work, there is more sodium on the outside of the cells and more potassium on the inside of the cell. Now, both of these are positively charged, but if you have a greater positive charge on the outside than you do inside, the difference in charge between those two locations is 70 millivolts. It's an actual electrical measurable amount. So we start with the sodium outside and the potassium inside. As the message moves through the neuron, those gated channels open much like you see the wave being done at a football game. Gates open starting at the dendrites and work their way down sequentially towards the cell body and then down the axon all the way to those terminal ends or boutons. The wave allows for the impulse to move through the cell. It does, however, result in all the sodium moving in, or at least an isotonic amount, and potassium moving out. Once that message has gone through, though, we have to reset. We do that using a pump. The sodium-potassium pump basically works to re-establish the original gradients. So if sodium is inside the cell, it will need to be pumped outside the cell. And the same with potassium only being pumped into the cell. One protein is responsible for connecting sodium and potassium on opposite sides and using ATP to sort of crank and reverse the locations. So the gated channels are a passive diffusion, and the sodium-potassium pump is an active transporting mechanism. Once that message has moved its way from the dendrites through the cell body and down the axons, that impulse, that electrical reversal charge, basically stimulates some exocytosis to occur here at the bouton. When the nerve impulse arrives, there are packages of neurotransmitters wrapped in membranes. Recall, those are called vesicles. The impulse causes those vesicles to move and fuse with the plasma membrane. That spills out the neurotransmitters into a space known as the synaptic cleft. Across that space, those neurotransmitters then move and attach to the receptors, locations where stimulus will be taken in, and the next neuron will begin its process. It'll begin with facilitated diffusion using those gated channels. Now, calcium is involved in this process because calcium is part of what stimulates those vesicles to fuse. This is one reason why neurons require a tremendous amount of calcium, not just your bones and teeth. Cells moving molecules in and out of their cells through that active process require ATP. That's the cellular energy. Well, we looked at active transport as well as endo and exocytosis. Although both endo and exocytosis, there are examples of passive as well as active processes. ATP is such an important molecule that we will spend a whole week, a whole unit on them. The ATP that fuels our cells is really a converted molecule, energy converted from the food we eat. That conversion is part of a larger set of what we call laws of thermodynamics. There are two laws that apply to biological systems. The first law of thermodynamics, also called the law of conservation, states that energy cannot be created or destroyed, only transformed and transferred, changed from one form to another, such as the food we eat transformed into the ATP molecules. The second law of thermodynamics, called the entropy law, really talks about as you convert that energy, there's always an incomplete conversion, a conversion in which forms of energy are, let's say, converted into forms that are not as useful, or maybe even lost to the organism. Entropy is that chaos that ensues if not enough energy is put into a system. For us, this conversion basically is applied in a way that, well, when we convert things, it's not 100%. And the forms of waste and heat and unusable other forms of energy that we lose, well, they play an Cells utilize two types of chemical reactions, and we name them based on the type of energy requirements. If a cell is going through a reaction in which energy is released, or the conversion releases a larger stored energy molecule into smaller ones, that is known as an exergonic reaction. In an endergonic reaction, we need energy put into the system. So either we're building larger things or storing energy. So it's an energy input. The two, exergonic and endergonic, are coupled together throughout our bodies. Think of a food that you eat being broken down through an exergonic reaction, making ATP, and then that ATP being used to actually put energy into a different molecule. So we're constantly switching between ATP stands for adenosine triphosphate. The adenosine is actually a complex molecule made of ribose and adenine. Recall that is a peptide and a simple sugar. The triphosphate is telling you how many phosphate groups are attached. If there were only two attached, we would call it adenosine diphosphate, and that is written as ADP. If we have three phosphates, then we have adenosine triphosphate, and that is written as ATP. The number of phosphates represents the amount of stored energy. So more phosphate groups, the higher the energy level. So ATP is the highest energy level our cells have for utilizing this small cellular energy molecule. They use that energy for all kinds of things in the cells, including chemical reactions, mechanical work, and even transporting things. The phosphate is transferred to reactants, to other things, to energize them. That's how it delivers its energy. Now in the diagram, you can see how ADP has a phosphate added to it to make ATP. That would require energy put into the system, and so which would that be, endergonic or exergonic? The reverse, ATP can be split apart, donating one phosphate to some energy requiring process, and in doing so is converted to ADP. Which is that, endergonic or exergonic? Enzymes work alongside with that ATP. Now think about that. We need to have energy in order to activate most of the cellular processes that need to be done. And that's kind of important because if energy wasn't required, things might just spontaneously occur when we don't want them to. So having an energy barrier, which is known as an energy of activation, that helps guide when and how chemical reactions will take place. We do that so that we can finite control them. But that also means we need not only the ATP, but the enzymes, who are the workhorse to getting everything done. Enzymes are made of proteins, and they help speed up reactions when we need them to take place. That is known as a catalyst. Now, enzymes are biological catalysts, and that's really important because we can make them, and they can be reused over and over. If in lab you recall us heating some substances, like Benedict's, well, heat is a catalyst as well, but it's not something we can recycle and use over and over. Enzymes are built in a very specific way. Remember, they're proteins. So we have a primary structure, secondary, and tertiary. And the point that we typically point out most of the enzyme's functionality, recall, is that 3D level, or the tertiary. Proteins are folded in such a way that there's a little groove where an actual chemical reaction will take place. We'll look at this more specifically in the next slide. Every enzyme has a fold or a specific area known as an active site. This is where chemical reactions take place. The chemicals that are going to be involved in the reaction are known as substrates. So substrates enter the active site of an enzyme, and in that active site, a reaction is catalyzed. Well, that might be a building or a breaking kind of reaction. So if we are doing dehydration synthesis and assembling things, enzymes might be involved in the building. In hydrolysis, where we're breaking things apart, enzymes would be there to help the splitting, and the associated ATP and water that's involved in both processes all take place right here at the enzyme. Enzymes are proteins, proteins that work at the structural level of a tertiary. Recall, the primary structure is really amino acid chains held together by very strong covalent or peptide bonds. But it is the secondary and tertiary levels and their folded up shapes that really give enzymes their functionality. These structural levels, however, are held together by much weaker bonds, things like ionic and hydrogen bonds. The shape in which an enzyme is folded gives what is called specificity. This is describing the actual shape of the active site and how it matches the substrate. This is much like a lock and key. The more teeth on a key to match a lock, the greater the specificity. So the more complex and exact a matching fit that the active site has for a substrate, the greater it'll have the confirmation of the two to join together. Enzymes work because of their shape, and if we do something to alter that shape, well, we alter their functionality. Specifically, if a condition has been subjected to this enzyme in which a weak bond is broken, well, we're going to change the ultimate shape of that active site. When you do that, that is called denaturing. Now don't mix up denaturing and hydrolysis. When you denature a protein, you alter its three-dimensional shape because you are breaking weak bonds. In hydrolysis, we are breaking those really tough peptide bonds basically all the way back to the amino acids or individual peptides. So denaturing happens when improper conditions, maybe a pH range that is outside what this enzyme was built to tolerate, could be temperatures, salts, heavy metals, all kinds of conditions can alter our enzymes and their functionality. One other category that can have an effect are inhibitors. Inhibitors are more like competitors. They're fighting for the active site with the real substrates. So as inhibitors, as their numbers get higher, there's more of them, they're generally going to outcompete the real substrate for the active site. The overall effect will be a slower rate of reaction. Now, the altering of these enzymes is something that will be critical in our coming up labs, especially the cellular respiration labs. We need to make sure that the critical enzymes that are involved in those processes are working. And if we subject those enzymes to an incorrect condition, such as a different pH, we might see how these enzymes are affected, becoming maybe non-functional or at least slowing or inhibiting them in some way. A key topic review for this chapter includes diffusion, both simple and facilitated, osmosis, the movement of water, and active transport. All of these mechanisms are for moving things in and out of cells. Recall that the passive transports require no energy, but do require a gradient, meaning a concentration difference. Because things are moving from high to low concentrations, a gradient is required. Active transport, on the other hand, is typically against a gradient, moving things from low to high concentrations. Whether it's moving down or up a gradient, all active transport mechanisms do require ATP energy. We also looked at endocytosis and exocytosis, moving of items, bulk or solids, in and out of the cells using vesicles. We used the neuron, the functional cell of a nervous system, to show how facilitated diffusion and active transport happen down the length of the cell, and then how exocytosis passes that message from one cell to the next. Lastly, we looked at energy, the thermodynamic laws, and enzymes. All of these are things you really need to review consistently, as we will be using them over and over throughout the rest of the term.